Hello friends, welcome to CEC live lectures. Dear friends, today in this session we are going to continue further in our series on cell biology. Today we are going to talk on the plasma membrane once again. Friends, this is the fourth lecture on the plasma membrane, and here we are going to talk once again on the functions of plasma membrane. We are specifically going to talk on active transport, and for the discussion on the topic we have with us in our studios Dr. Charu Dogra Rawat. Dr. Charu Dogra Rawat is Assistant Professor in Department of Zoology, Ramjas College, University of Delhi. Dr. Charu Dogra Rawat is a prolific professor, and she believes in giving her utmost knowledge to the students so that you are benefited as a whole. Friends, if you have any questions regarding the lecture, then you can call us through our toll-free number. Our number is 1-800-110-430. I repeat, our number is 1-800-110-430. You are requested to call in the last 10 minutes of the lecture so that first you could have deep insight into today's topic, that is active transport, and later on, of course, we'll try to give answers to your questions in the last 10 minutes. Now, I would like to welcome our guest, Dr. Charu Dogra Rawat. Hello, ma'am. Welcome to the lecture.
So continuing with the series on cell biology, as Gitika said, plasma membrane we'll be talking about, and the learning objectives for this session particularly would be to understand the functions of the plasma membrane. We have been talking about the different functions of plasma membrane, particularly related to the transport, and we have talked about different mechanisms of transport across the cell membranes or plasma membranes. Today we'll be talking about the active transport, the various forms of active transport and various carrier molecules which causes this active transport to take place. Most of the figures etc. are taken from CARP, Cell and Molecular Biology, Concepts and Experiments 7th edition, wherever they are not mentioned, otherwise it will be mentioned along with. So the mechanisms of movement of substances across membranes, we have seen that there are 4 different methods which constitutes the passive as well as the active transport. Passive and active difference is basically for the utilization of energy. In the passive transport, there is no utilization of energy because, as you can see, the transport is basically along the concentration gradient. So you can see these two types are the non-mediated ones, the A and the B. The larger, the capital, the bigger letter basically means that the concentration is higher on this particular side and the concentration is lower on this side. So the non-mediated one, which basically means that they do not require any specific proteins across for passing this ions or molecules across the membrane, and the transporter mediated one. So the 4 different mechanisms are the simple diffusion through the bilayer. So certain molecules or ions basically they just diffuse. Usually they are very hydrophobic molecules because this lipid bilayer is hydrophobic. So the molecules they can transport across the concentration gradient from one side to the other by just mechanism simple diffusion, and we have already discussed about that. The second was basically the simple diffusion through an aqueous channel. So these are the hydrophobic molecules because they can pass through this lipid bilayer, which are hydrophobic molecules, but hydrophilic molecules there are certain carrier proteins which are called as the aquaporins. So there are certain transmembrane proteins which are already present, although it is a non-mediated one because it does not require any carrier molecule to cross it across the membrane. So again, this is towards along the concentration gradient for the hydrophilic molecules to the aqueous channels or the aquaporins. Other transport again, which is passive transport, the third transport mechanism was the facilitated diffusion, and in that there are certain important carrier proteins which carry the molecules from one path to the other, but this is again the concentration along the concentration gradient from the higher concentration to the lower concentration. The fourth mechanism which we will talk about today is the active transport, where it is basically against the concentration gradient. As you can see, the D is bigger here and smaller here, which means that it's highly concentrated here and a lower concentration is here. So for moving against the concentration you need energy, and this energy is provided by the hydrolysis of certain energy molecules such as the ATP. So active transport by means of a protein transport, so it is a transporter mediated, it is an active transport molecule, active transport mechanism, and it needs the hydrolysis of ATP molecules. So we will talk about in detail this fourth transport mechanism for the movement of substances across the plasma membrane. Active transport, so why do we need, or when we talk about the passive transport mechanisms, the purpose is basically to equalize the concentration on both the sides. When the movement is from the higher concentration to the lower concentration, so basically the movement is towards the equilibrium. So the concentration of the solutes or the molecules or the ions become constant on both the sides of the plasma membrane. But in a cell, the life just cannot exist under these equilibrium conditions. If we look at the ion concentrations inside and outside of a typical mammalian cell, which is presented in this table, you can see that the sodium ion concentration, for example, extracellular concentration is 150 mm, whereas intracellular concentration is 10 millimolar. So there is an ionic gradient which is there, and this is a 15 times, the gradient is there. Similarly, the potassium gradient, it's an opposite kind of a thing. The extracellular concentration is low, which is around 5 millimolars, and the intracellular concentration is much more, which is 140 millimolars, which is 28 times more than what is extracellularly present. Chloride ions, calcium ions, hydrogen ions, you can see at the pH of 7.4, there is a concentration of 10 raised to the power of minus 7.4 molar extracellular concentration. However, the inside the pH is at pH is 7.2, which is 10 raised to the power of minus 7.2 molar, which is nearly 2x. But some of the molecules, as you can see, the calcium molecules are 20,000 times more towards one side or on the other side. So life is not just existing in a cell under equilibrium conditions. The ionic concentrations across the plasma membranes is not equal. They are different. Either intracellularly or extracellularly, they are maintained at a higher or a lower concentration. So this maintenance of the lower and higher concentration is because of this active transport of the molecules, as we will see. The ability of cell to generate such steep concentration gradients across its plasma membrane occur by active transport. Like facilitated diffusion, which we talked about, active transport depends on the integral membrane proteins. Therefore, it is a transporter mediated transport that selectively bind a particular solute and move it across the membrane in a process driven by the changes in the protein's conformation. So it is very similar to facilitated diffusion, where there are certain specialized molecules called as the transporter molecules, the change in the conformation of which drives the movement of the molecules from one side to the other. So it's just like facilitated diffusion. However, it differs from the facilitated diffusion because the movement of the solute is against the concentration gradient and requires to be coupled with the input of energy. So as we saw, the ATP hydrolysis needs to take place. The energy needs to be spent for the movement of the solutes against the concentration gradient. The androgenic movement of ions or other solutes across the membrane against a concentration gradient is coupled to an exergonic process. So androgenic is basically, what we meant by that is that it is requiring energy, it is consuming energy. ATP hydrolysis is there, so it is consuming energy, but usually it is associated, it needs to be coupled to an exergonic process such as the hydrolysis of ATP, the absorbance of light, the transport of electrons, or the flow of other substances down the gradients. So we will see as we subsequently move into the session that this particular androgonic movement, because it is consuming the energy, so alongside there must be something which is forming the energy or releasing the energy is there. So it is a coupled process, and this formation of the energy or release of the energy is basically coupled by certain mechanisms such as the hydrolysis of ATP or absorbance of light or transport of electrons, etc. The proteins that carry out this active transport, because it is a transporter mediated protein transport, therefore these proteins that carry out the active transport are often referred to as pumps, which is there. So we will talk about the primary active transport first, which undergoes the coupling of the transport to the ATP hydrolysis. In 1957, a Danish physiologist by the name of Janus Kuh discovered an ATP hydrolyzing enzyme in the nerve cells of a crab that was only active in the presence of these two ions, the sodium ions and the potassium ions. This enzyme was called as the sodium-potassium ATPase or the sodium-potassium pump. So we will talk in detail about the mechanism of action of these sodium, the structure, as well as the action of these sodium-potassium pumps, because they are very important to understand this active transport which is there. It is the sodium-potassium ATPase that is responsible for the large excess of sodium ions outside of the cell and the large excess of potassium ions inside the cell. So, we just saw that there is a disequilibrium in the distribution of sodium and potassium ions across the cell membrane. Sodium ions are much higher in concentration, around 15 times higher in concentration outside the cell than inside, whereas the potassium ions were much more inside the cell, around 28 times higher inside the cell than outside. So, how is this disequilibrium maintained? So, sodium-potassium ATPase are the proteins which are responsible for maintaining this disequilibrium or the difference in the concentration across the membranes, and we will just see how it is done. So, you can see that for electrical neutrality across the membrane. So, if you see this particular diagram, you can see that this is the plasma membrane or a cell membrane, this is the extracellular part and this is the intracellular part. So, in the extracellular part, you can see there are sodium concentration is high. So, you can see that there are pentagons, the sodium ions are much more in number than what they are inside. Inside, the potassium concentration is much higher. So, these purple squares are the potassium ions which are much higher than what they are extracellularly. And because of this, the positive is in here, it is much more, and here there has to be some electrical neutrality which needs to be there. And this positive which is outside is neutralized by the presence of the chloride ions. So, you can see that the chloride ions are much more in number extracellularly than what they are inside, and inside the electrical neutrality is maintained by these orange rectangles, which are basically the membrane impermeable anions that arise from a variety of sources including the proteins. So, for electrical neutrality across the membrane, chloride ions are present at greater concentration outside the cells where they balance the extracellular sodium ions, and the abundance of intracellular potassium ions is balanced primarily by excess negative charges carried by the proteins and nucleic acids. So, in that way, basically some kind of neutrality is maintained. However, still there is a difference in the charges which are, and these sodium-potassium ATPases maintains the difference in the charges across the membrane, but they try to neutralize this excess of one or the other ion present in the outside or the inside of the cell. This particular thing represents the sodium-potassium ATPase, and it is just showing the movement of the potassium, which is basically outside. With an arrow represents a transmembrane potassium channel and the direction of the net potassium movement. So, the direction of the net potassium movement should be towards the outside, which is there. So, this is how the membrane resting potential looks like. The ratio of sodium-potassium pumped by the sodium-potassium ATPase is 3:2. For each ATP hydrolyzed, 3 sodium ions are pumped out as 2 potassium ions are pumped in. So, we already know that outside the sodium concentration is too high, but the sodium ions are still thrown out by utilizing the energy from this ATP. So, ATP is hydrolyzed, and in the process 3 sodium ions are thrown out and 2 potassium ions are taken in. Inside there is already a high potassium concentration. So, this movement is actually against the concentration gradient which is there, and the movement of sodium is against the concentration gradient because outside there is already high sodium concentration which is present. So, the ratio is not 1:1 but 3:2. So, 3 sodium for each ATP molecule hydrolyzed is thrown out and 2 potassiums are taken in, which is there. So, sodium-potassium ATPase is electrogenic, which means that it contributes directly to the separation of charge across the membrane. So, as I said, that electrical neutrality is tried to be maintained by the chloride ions as well as the negative ions, which are negative impermeable negative ions, which resources are the proteins and the nucleic acids. But because of this pumping of the sodium and potassium ions, the electrical non-neutrality or the charge difference across the plasma membrane is always maintained, which is there. So now we will look at how this sodium-potassium ATPase works. What is the simplified schematic model of the transport cycle? The step 1 is that this is the sodium-potassium ATPase, a protein molecule, and it is said to be in a particular conformation which is termed as, for example, E1 conformation. So this is a particular conformation of sodium-potassium ATPase, and as you can see, this is the extracellular environment, and we will walk through all these 8 steps. So these are the extracellular, this is the cytosol which is there, and as you can see, this in this particular conformation, the face which is towards the cytosol side is open. However, the conformation which is towards outside, the face towards the outside is closed. So the step 1 is basically that the pump is in the E1 conformation and the ion binding sites are accessible towards the inside of the cell. In this step, the protein has bound 3 sodium ions and an ATP molecule. So in this particular conformation, the ATP molecule is bound and the 3 sodium ions are bound onto the ion binding sites towards the inside of the cell. In the step 2, a gate within the protein has closed. So this gate has been closed, or it has been this channel has been occluded, shifting the pump into an occluded even state in which the sodium ions can no longer flow back into the cytosol. So these sodium ions are in concentration here. So basically what happens is that they bind to these ionic binding sites, and then this particular gate closes, and therefore they cannot now flow back, and the ATP is also bound. In the step 3, hydrolysis of the bound ATP take place. So this ATP gets converted into ADP and PI, and the energy is produced. This energy causes a change in the conformation of this protein molecule. So in step 3, in this particular state, the ATP hydrolysis has occurred and ADP and PI are formed, and the conformation has changed from E1 to E2 conformation which is there. In the step 4, the release of ADP causes the change from the E1 to the E2 conformation. In doing so, the binding site becomes accessible to the extracellular compartment, and the protein loses its affinity for the sodium ions, which are then released outside the cell. So you can very clearly see here, when the ATP hydrolysis has occurred, the conformational change has occurred in the protein molecule, and this E2 conformation, this particular side has been opened, and therefore the affinity for the sodium also reduces to the ionic binding sites, and the sodium are just released. Now this channel has opened, so sodium is just released. So the binding affinity of this molecule changes from sodium to potassium. Now potassium ionic bind sites, potassium ions has more affinity to that. So in this particular conformation, the 2 molecules of potassium, there are 2 binding sites for potassium. So the potassium binds to the ionic sites in this particular conformation. So that is with the step 4. So once the 3 sodium ions have been released, the protein picks up the 2 potassium ions. So the protein has now picked up the 2 potassium ions, after which this particular channel which were opening towards the extracellular now closes, or it is occluded. As you can see again in step 6, the closing of another gate within the protein shifts the pump into an occluded state, thus preventing backflow of the potassium ions. So now these potassium ions, because the specificity is more, the binding is more, as well as the gate is closed, these 2 potassium ions cannot now flow back. Because these potassium ions cannot flow back, they just remain bound to this particular site, and again the conformation changes. So the dephosphorylation occurs. Now what happens is, because ATP has ADP is being formed, so PI is released and this particular molecule is phosphorylated. Now this transporter will again be dephosphorylated, and the ATP will be formed. So in step 8, ATP binding induces the return of the protein to the original even conformation, and in this even conformation, as you can see, that the face towards the intracellular side has now opened, and therefore the 3 sodium molecules can bind to their ionic sites, and the entire process is again repeated. So what happens is that there are 3 important things which are to be remembered for this particular mechanism of action. First is that sodium ions, they bind only to the protein molecules when they are in even conformation state. Potassium ions only bound to the protein molecule or the sodium potassium ATPase when it is in the E2 conformation change. Because of this E1 to E2 conformation change, the binding affinity of the sodium potassium ATPase towards sodium ions or potassium ions differs. So these are two important points. Third important point is that the change in the conformation is catalyzed by the phosphorylation state of the protein and the hydrolysis of ATP molecule. So when the ATP is bound to the protein, it is in even conformation, but when it gets hydrolyzed, then the conformation changes to E2. So this E1 to E2 conformation is occurring because of the ATP hydrolysis, and when the dephosphorylation occurs, the E2 conformation protein comes back to the even conformation state. So when it comes back to the even conformation state, sodium ions can be bound, conformation change occurs, it opens towards outside, affinity to sodium reduces, affinity to the potassium increases in E2 conformation, potassium binds, dephosphorylation occurs, and E2 comes back to the even conformation, and this cycle is again repeated. So this particular distribution of or the transport of sodium and potassium ions across the membrane maintains the disequilibrium of sodium and potassium ions across the membrane. The sodium ions, the 3 sodium ions are thrown to outside the cell where the concentration is already high, and the potassium ions are taken up inside the cell where the concentration is already high for the potassium concentration. So that is how basically the sodium potassium ATPase works to maintain this difference in the concentration of sodium as well as potassium ions across the plasma membrane. The sodium potassium pump is found only in animal cells. This protein is thought to have evolved in primitive animals as the primary means to maintain cell volume and as the mechanism to generate the steep sodium and potassium gradients that play such a key role in the formation of impulses in nerve and muscle cells. We will not go into detail about the mechanism of action of nerve and muscle cells, but you have very clear idea that in nerve cells the propagation of the nerve impulse needs to take place, and this propagation is basically in the form of the electrochemical gradients which is generated. So this propagation is facilitated by the electrochemical gradient which is maintained by the distribution of the sodium and potassium ions across the membrane. Similarly, in muscle cells, the contractibility and the decontractibility is because of the distribution. One of the reason is because of the distribution of the ions across the muscle membrane. So this is again maintained by the sodium potassium ATPases, which causes the difference in the ionic or contains a kind of an electro gradient, because of which the muscle contraction or the nerve impulse transport can take place. So we generally that detail about the action of this nerve impulse and muscle cell is taken up in physiology, but the root base for that is this gradient which is generated by the sodium potassium ATPase molecule across the cell membranes of nerve and muscle cells which is there. The importance of the sodium potassium pump becomes evident when one considers that it consumes approximately 1/3 of the energy produced by most animal cell and 2/3 of the energy produced by nerve cells. So you can imagine how important this pump is, because 1/3 of the energy or ATPs which a cell is producing is consumed for the working of these sodium potassium ATPase molecules. So this sodium potassium ATPase, which maintains the electrogradient across the plasma membrane, is very, very significant and important, because the cell directs 1/3 of its energy towards this process only. Sodium potassium ATPase is an example of a P-type ion pump. Now what does this P-type ion pump means? The P stands for the phosphorylation, indicating that during the pumping cycle, the hydrolysis of ATP leads to the transfer of the released phosphase group to an aspartic acid residue of the transport protein. So when we were going through the mechanism, we just saw that when the ATP hydrolysis is occurring, then the phosphate group is actually moved to the transfer protein only. The transport protein is in E2 conformation, is in the phosphorylated state. From E2 conformation to even conformation, it can come only if the dephosphorylation occur. So the pumps in which the transport protein is phosphorylated are said to be P-type ion pumps, and sodium potassium ATPase is an important type of this particular P-type ion pump which is there, because the transport protein itself is phosphorylated taking up the phosphate group from the ATP hydrolysis which it is releasing. There are other kind of primary ion transport systems also. The best studied p-type pump is again a p-type pump is a calcium ATPase present in the membranes of the endoplasmic reticulum. So we have seen that sodium potassium ATPase is abundant in the nerve cells as well as in the muscle cells. In the plasma membrane of endoplasmic reticulum, calcium ATPases are also present, where it actively transports calcium ions out of the cytosol into the lumen of this particular organelle. So again you can see here, this is the calcium, the cytosol, and this is the lumen. So in the cytosol the concentration is less, and in the lumen the concentration is much higher. So this particular triangle reflects that the concentration is low here. So against this concentration gradient, the calcium is moved from the cytosol, is taken up by the endoplasmic reticulum plasma membrane through this transmembrane protein or through this transporter protein which is termed as the calcium ATPase. So this is the calcium ATPase, again a similar mechanism. There is an even conformation. During this even conformation, there are two sites which are present here, and they have high affinity towards the calcium binding, and this is the ATP site. So the calcium will be bound here, ATP will be bound here. In the next step, the ATP hydrolysis will take place. This hydrolysis will cause the change in the conformation, and because of the change in the conformation, you can see that the inside will go towards outside, and these will now become the low affinity calcium binding sites, and in this particular conformation, the calcium will be released inside the lumen. And as you can see, this transport membrane itself is phosphorylated. That is why it is a p-type ion pump. Then the dephosphorylation will occur. The dephosphorylation will cause it to come back into the even conformation, and there will be again two high affinity calcium binding sites as well as a ATP binding site, and this again the cycle will be repeated. So typical p-type ion pump which is present in the endoplasmic reticulum plasma membranes, and it is termed as the calcium ATPase which is there. Important steroids or the drugs are there which produces their effect by binding or by nullifying the effect of these sodium potassium ATPases which is there. So you can see an important steroid obtained from the foxglove plant is called as digitalis, and it has been used for 200 years as a treatment for congestive heart disease. What it does, it binds to the sodium potassium ATPase. Digitalis actually strengthens the heart's contraction by inhibiting the sodium potassium pump, which leads to a chain of events that increases the calcium availability inside the muscle cells of the heart. So for increasing the concentration, the calcium accumulation needs to take place. And how does this happen is shown in this particular diagram, or the action of a particular drug is shown here. So you can see this is a sodium potassium ATPase which is there, and the sodium is moving out and the potassium is moving in. This digitalis actually inhibits the action of this sodium potassium pumps, due to which there will be an increased sodium concentration inside the cell. We will talk about the sodium calcium exchanger in a while when we talk about co-transportation, but for here we can just understand that the increase in sodium concentration will increase in relative concentration of calcium ions inside the cells. This is because of this sodium calcium exchanger. The higher concentration will lead to the movement of calcium ions also inside, and the calcium ions, this is a kind of a co-transportation. So when the sodium is higher inside, the calcium will be higher inside, and the increase in calcium increase will lead to the higher troponin binding, the calcium binding, and will therefore increase the contractibility of the cardiac cells. So in the heart contraction increases. So this particular compound is actually acting on this sodium potassium ATPases. So it again reflects the importance of sodium potassium ATPases, the function of sodium potassium ATPases in the transfer of or maintaining of the differences in the concentration of sodium as well as potassium ions across the membrane, and how inhibiting such ATPases can lead to different actions such as the higher contraction or inotropy of the cardiac cells which is there. So sodium potassium, so all these p-type ion pumps are also present in the plasma membrane, and this is one mechanism of active transport which we study.
With this note, thank you, ma'am. Thank you so very much for giving us this session. Friends, you are requested to be with us as we are back after a short break. Thank you. Hello friends, welcome back to this session. Friends, as you know that today we are talking on function of plasma membrane, and specifically we are discussing on active transport. For the discussion on the topic, we have with us in our studios Dr. Charu Dogra Rawat. Dr. Charu Dogra Rawat is a prolific professor. Through her, we always get in-depth knowledge on various topics and issues of zoology. Friends, if you wish to ask questions on today's topic from Dr. Charu Dogra Rawat, then you can call us through a toll-free number. Our number is 1-800-110-430. I repeat, our number is 1-800-110-430. Now I would like to welcome our guest, Dr. Rawat, once again. Hello ma'am, welcome to the lecture.
So continuing with the active transport and continuing about talking about the various pumps, we have already talked about sodium-potassium ATPase, which is a p-type pump. So p-type ion pump, I repeat, is basically the one where during the pumping cycle, the hydrolysis of ATP leads to the transfer of the released phosphate group to an aspartic residue of the transport protein. So the transport protein itself gets phosphorylated during this particular cycle, and therefore they are termed as the p-type ion pumps which are there. Now plant cells have a hydrogen transporting p-type plasma membrane pump. In plants, this proton pump plays a key role in the secondary transport of solutes. So this is just another example of a p-type plasma membrane pump which are present in plant cells, and they are the hydrogen ion transporting cells. In plants, this proton pump plays a key role in the secondary transport of solutes in the control of cytosolic pH and possibly in control of cell growth by means of acidification of the plant cell wall. So you can see here that this is the hydrogen or the proton pump, as it is said, a proton ATPase which is present, and it causes the uptake of the hydrogen ions, although there is a more positive side which is present here. So this plays a key role in the transportation of the or increase of the decrease of the cytosolic pH which is there. So the pH is 7.5 and this pH 5.5. So along with it causes a change in the nutrient and the osmolyte uptake, and therefore you can see that, for example, the wall is loosening or the cell wall is loosening here. So again, this H plus ATPase or the proton ATPase has a very important role, and the process of transport will involve this particular ATPase molecule to be phosphorylated. So the transporter protein will be phosphorylated, and therefore it is termed as the p-type plasma membrane. Another important p-type pump is the proton potassium pump ATPase, which is present in the epithelial lining of the stomach. So the epithelial lining of the stomach contains this H plus K plus ATPase, which secretes a solution of concentrated acid up to 0.16 normal HCl into the stomach chamber. So we all know that stomach is acidic in nature, the content of the stomach, and there are certain cells in the epithelial lining of the stomach which releases this acid into the stomach for the digestion to take place, and the release of this acid into the stomach by these parietal cells is through these particular ion channels, which are called as the H plus K plus ATPase. In the resting state, these pump molecules are situated in the cytoplasmic membranes of the parietal cells of the stomach lining. So you can see that this is, these are the parietal cells. This is a histological section. In the parietal cells, you can see that these particular cells are lying, and these are the inactive hydrogen potassium ATPase molecules, and they are situated near the cytoplasmic membrane of the parietal cells, and they are non-functional. Now, when there is a there is a stimulus, for example, by the binding of this particular hormone, which is the histamine. So if the histamine binds here, these hydrogen potassium ATPases will go and they will start binding towards these villi, and they will become active. So what happens in here is, when food enters into the stomach, a hormonal message is transmitted. So when the food is there, this is the hormone, and this hormonal message basically transmits the information, causing these hydrogen potassium ATPase molecules to move to the apical cell surface, where they fuse with the plasma membrane and begin secreting the acid. So you can see again in this diagram, and this is the zoom out session where it just shows that this hydrogen potassium ATPase has now come to lie here and fuses with the plasma membrane, and this has caused the hydrogen ions, which is basically the acid which is there, the acid to be thrown out and the potassium ions are taken in. So the hydrogen ions start moving outside. These red arrows show that the drugs which we take, there are certain drugs which are acid blocking drug. They will block the receptors where the hormones are bound. Then there are certain agents which will cause the excess hydrogen ions to be neutralized by the hydroxyl ions and will decrease the effect of the acids. But there are certain drugs which are pump inhibiting drugs, so they will bind to the pump itself and stop this increase of the acid. Because when the food enters, the acid needs to be released into the stomach for the proper digestion of the food. But if there is too much of an acid, it causes acid reflux, and therefore we have to take certain drugs. And we will see that what is the mechanism of some of these drugs which take place. So this is the entire working of the or the stimulus which is given, and then how the hydrogen potassium ATPase works, which are present in the parietal cells of the epithelial lining of the stomach, which is there. In addition to functioning in digestion, as we said, the stomach can also lead to heartburn. When there is an excess production of acid, then there is an acid reflux which occurs, or there's a condition, physiological condition, which is termed as heartburn. And we take certain drugs, for example, the Prilosec or the related drugs, which are widely used to prevent heartburn, and they act by inhibiting the stomach H plus K plus ATPase. So you can see here that this is the proton pump. In the previous diagram, we just saw the action of one hormone which is there, but there are other hormones such as gastrin, acetylcholine and histamines, and these hormones stimulate the production of HCl. But when we take this Prilosec and other related drugs inhibits the action of this proton pump, and therefore the acid is not released. Therefore, they give you the relief from the heartburn which is caused by the excess acid production. So, irrespective of whatever the stimulus is there, these drugs bind or act by inhibiting these hydrogen potassium pumps which are present in the epithelial lining of the stomach. There are other acid blocking heartburn medications, for example, Zantac, Pepcid and Tagamet. They do not inhibit the hydrogen potassium ATPase directly, but block a receptor on the surface of the parietal cells, thereby stopping the cells from being activated by the hormonal stimulus. So these are certain other drugs will bind to these particular receptor molecules inhibiting the binding of the hormones and therefore again production of the acid or release of the acid into the stomach. But certain drugs actually act on these particular ATPases. So why I am talking about these particular drugs is because it substantiates the role these particular ATPase molecules, ATPase proteins play in in causing certain functions such as the release of acid in here or in impulse propagation, etc. So the effect of drugs tells us that if that these particular ATPase molecules are playing very significant role in different plasma membranes. Apart from P-type pumps, there are one more type of pumps which are called as V-type pumps. V is because of the vacuolar thing. So they utilize the energy of ATP without forming a phosphorylated protein intermediate. They are large multi-subunit complexes and similar in structure to that of the ATP synthase. V-type pumps actively transport hydrogen ions across the walls of cytoplasmic organelles and vacuoles, and therefore they are termed as the vacuolar type. So you can see this is the V ATPase, and this V ATPase is a multi-subunit protein. It has a catalytic hexamer. That catalytic hexamer is because of where the ATP will bind, and therefore hydrolysis of ATP will take place. This hydrolysis, however, will not lead to the phosphorylation of any of these proteins. Therefore, it is a V-type protein. This is kind of a stalk, and then this is the proton pathway. Primarily, these V-type pumps are involved in the movement of the protons or the hydrogen plus H plus ions, whereas the sodium-potassium ATPase pumps or calcium ATPase pumps, all P-type pumps are involved in the transport of various other ions than just protons. So these are the V-type ions which are present. These V-type ions, ion proteins, they occur in the membranes that line lysozymes, secretory granules and plant cell vacuoles, where they maintain the low pH of the contents. V-type pumps have also been found in the plasma membrane of a variety of cells. For example, the V-type pump in the plasma membrane of kidney tubules helps maintain the body's acid-base balance by secreting protons into the forming urine. So this is just an example of a V-type pump which is given here, which is present in the renal alpha intercalated cells. So you can see this is the proton pump which is there, and this proton pump causes the release of the hydrogen ions into the urine. In conjunction to these cells can also absorb a carbon dioxide and release HCO3 minus through a chloride hydrogen bicarbonate exchanger, which we'll talk about subsequently, thus maintaining the pH homeostasis in the blood. So this is the blood and this is the urine. So excess hydrogen ions are released into the urine, maintaining the acid-base balance of the blood, which is very important. And this again you can see is the lysozyme. So in the lysozymes, these proton pumps are also present. These proton pumps will keep on pumping hydrogen ions inside the lysosome, again maintaining the pH at very low. So lysozyme, we know that contain lot of acid hydrolysis, and it's kind of a suicidal bag which is involved in the digestion or the degradation of various molecules. So the pH inside the lysozymes are very low, and this is maintained by these V-type ion pumps or the proton pumps which are present on its surface, pumping hydrogen ions inside the lysozyme, maintaining an acidic environment. So similarly, the urine, the excess hydrogen ions is released into the urine maintaining the homeostatic balance or acid-base balance of the blood which is there. So again, V-type ion pumps are very, very important. The third type of ion pumps which are present, they are said to be ATP binding cassettes or ABC transporters, so called because all of the members of this superfamily share a homologous ATP binding domain. So this is the structure of a typical ABC full transporter. So you can see that this is the typical structure of a ABC full transporter, and it contains two TMDs or a transmembrane domains, TMD1 and TMD2. Each domain has 5 to 6 transmembrane proteins which are joined together. Apart from that, there are two NBDs, which are termed as the nucleotide binding domains, which are there, each containing, so these are the two NBDs which are there. One is shown in the red and one is shown in the green. This is towards the inside or the cytosolic side of the membrane. Towards the outside, it's basically the transmembrane protein which interacts with it. So this is a typical protein. There can be a half ABC transporter which contains just one TMD and one NBD. So there is a whole variety of ABC transporters which exist, but this is the structure of a full transporter, and this domains are very much conserved in all these receptors, and they contain a very homologous ATP binding domain which is present. I'll take an important example of this ABC transporter, which is the cystic fibrosis transmembrane conductance regulators, or CFTR, which is an ABC transporter which is there. Now, cystic fibrosis is the best studied and most common inherited ion channel disorder that results from a defect in the ion channels of the epithelial cell. Although cystic fibrosis affects various organs, so there is a mutation in a particular gene which leads to cystic fibrosis, and this gene is for this particular CFTR receptor which is there. So this is the ABC transporter, and a defect in this particular regulator leads to the congenital disease, which is the cystic fibrosis. Although cystic fibrosis affects various organs including the intestine, pancreas, sweat glands and reproductive tract, the respiratory tract usually exhibits the most severe effect. The victims of CF produce a thickened, sticky mouth that is very hard to propel out of the airways. Afflicted individuals typically suffer from a chronic lung infection and inflammation which progressively destroy the pulmonary function. So, respiratory tract is the or the airways are the most effective or most affected organs in the cystic fibrosis patient, and we will see that how this ABC transporter dysfunctioning leads to this particular symptoms in the cystic fibrosis patients. You can see this is the airway epithelium from a normal individual. So, in a normal individual there is an epithelial lining which is there, and there is a normal exchange which is taking place. The CFTR is responsible for the transport of the calcium of the chloride ions. So, you can see that the chloride ions are basically moving across the cell membrane. This is the transporter. So, chloride ions are moving outside, sodium ions are moving inside, water is moving outside. These are the epithelial cells. So, this is the mucus layer of the epithelial cells. It is well hydrated because of the movement of the hydrogen, and therefore the bacteria is suspended and it keeps flowing out. It does not stay, and the bacteria is just keeping flowing. In the airway epithelium of a normal individual, water flows out of the epithelial cells in response to the outward movement of ions, thus hydrating the surface mucus layer. The hydrated mucus layer with its trapped bacteria is readily moved out of the airways, keeping the airway clean and in a very and the individual healthy. What happens in a cystic fibrosis patient? In the airway epithelium of a person with the cystic fibrosis, the abnormal movement of the ions causes the water to flow in the opposite direction. So, you can see here that the CFTR protein is not working, or it's not there, it's dysfunction because there is a mutation in the gene. CFTR protein is not produced. So, because of the CFTR protein is not there, the chloride ions cannot move out. Because the chloride ions can't move out, the water also can't move out, and there is an uptake of the water causing this mucus layer to be dehydrated. Because of the dehydration of the mucus layer and there is deficient flow of the bacteria outside, the bacteria starts accumulating on this epithelial cell lining. They create a kind of a biofilm which is very, very dangerous. So, as a result, the trapped bacteria cannot be moved out of the airways, which allows them to proliferate as a biofilm and causing the chronic infections which are there in the airway epithelium. So, that is the again reflecting the significance of these pumps which are present in the plasma membrane. So, because of the defect of these ion pumps or ABC transporter, which is a type of ABC transporter, the CFTR regulator protein, because of the defect in that, the cystic fibrosis disease is occurring, not because there is a impaired movement of ions across the plasma membrane which is there. Apart from these primary active transport which we are talking about, which is the result of the direct ATP hydrolysis. So, we have talked about three different primary active transporter pumps.
which are the P-type pumps, V-type pumps and the ABC transporters. So sodium-potassium ATPase is the example of P-type pump, hydrogen-proton ATPase is the example of a V-type pump, and CFTR is an example of a ABC transporter, which we have covered. So all these primary active transport mechanisms involve the hydrolysis of the ATP. Whether the transporter protein is phosphorylated or not, that is not important, but as far, that is a different thing. But what we have seen is that the primary active transport involves directly the hydrolysis of ATP. There is also something which is called as a secondary active transport or co-transporter or anti-transporter, which we will see. So in this secondary active transport, because of the ionic difference that is maintained by the primary active transporters, the transport of certain molecules across the plasma membrane takes place, which directly does not involve ATP hydrolysis, but because they are electrogenic processes or exogenic processes, they maintain this electrical differences across the plasma membrane that causes the secondary transport to be there. So we will see into the detail about these this co-transport and what are the different mechanisms of this secondary active transport which are there. So this secondary active transport is coupled to the transport to the existing ion gradients. The establishment of concentration gradients such as those of sodium, potassium and protons provides a means by which the free energy can be stored in a cell. The potential energy stored in these ionic gradients is utilized by the cell in various ways to perform work, including the transport of other solutes against the concentration gradient. So this is just one of the example by which the ionic gradient which is created by the primary active transport and the energy which is the free energy which is stored in the cell is utilized. So this is utilized for the transportation of other solutes, other molecules or other ions across the plasma membrane also, and therefore it is termed as the co-transport mechanism. Co-transport mechanism can be of two types. So one term which is used is termed as the uniporter. So uniporter is basically when only one molecule is transported. So amino acids, nucleosides, sugars, they just transport across the plasma membrane as single molecules. But sometimes there are two molecules which are transported through the same transporter protein. These two molecules can be transported towards the same site, inside or outside. That is termed as a symporter or a co-transporter, the movement in the same direction. We will see in detail the example, the sodium glucose co-transporter in the intestine. And then there is a term which is called as antiporter or the exchanger. Antiporter or exchanger means that the direction of the movement of the two molecules through the transporter is in the opposite direction. So one is coming to one direction and the other is going to the other direction, and it is termed as the antiporter or the exchanger, and we will see the chloride bicarbonate exchanger in the red blood cell is the type of an antiporter. So starting with the sodium glucose co-transporter, we will see that the sodium concentration we know is kept very low within the cells by the action of a primary active transport system. So we have already seen the working of sodium-potassium ATPase, and this sodium-potassium ATPase is responsible for maintaining low concentration of sodium inside the cell. This is located in the basal and lateral plasma membrane, which pumps sodium ions out of the cell against a concentration gradient. So we have already seen that. Now the tendency for sodium ions to diffuse back across the apical plasma membrane down their concentration gradient is tapped by the epithelial cells to drive the co-transport of glucose molecules into the cell against a concentration gradient. So what you can see here, this is a sodium-potassium ATPase, and this sodium-potassium ATPase is responsible for maintaining the low concentration of sodium outside. Now there are certain other transporter molecules which are present. Because of the low concentration of sodium inside, now this molecule has a tendency that the higher sodium molecules will just diffuse inside. The low concentration will propel this to happen passively. Now this, when it can happen passively, this co-transporter uses this passive diffusion or the facilitated diffusion, which is a passive mechanism for the glucose molecules also to move inside against the concentration gradient. So this movement of sodium molecule is along the concentration gradient. Because there is a lower concentration inside, This transporter is facilitating the movement of the sodium ion inside, along the concentration gradient, but the cell utilizes this by moving the glucose molecules inside against the concentration gradient also. So this sodium glucose transporter moves 2 sodium ions and 1 glucose molecule with each cycle. So this is termed as the cotransporter. The maintenance of this sodium disequilibrium causes the sodium influx. So sodium influx is passive. So this sodium influx is it collaborates with the glucose molecules, or there are certain transporter which collaborates the movement of the sodium inside, sodium influx with the glucose influx also against the concentration gradient. So this is an example of a co-transporter or a sim transporter which is there. Antiporter or exchangers which we are talking about. So there are, for example, 3 examples I have given in here. The antiporters or the exchangers. One is a sodium calcium exchanger, or NCX it is termed as. These are present or important in muscle cells, and they maintains the low intracellular concentration of calciums. So you can see 1 calcium moves out. So 3 potassium, the potassium has the tendency to move inside the cell because the inner sodium concentration is lower. So when the 3 sodium ions move inside, through this transporters 1 calcium just moves outside. So this particular passive diffusion is causing the 1 calcium ions to move outside. So this is an exchanger or an antiporter which is present. Another one is present for maintaining the proper cytoplasmic pH by coupling the inward or downhill movement of sodium with the outward movement of H+. So this again, sodium is responsible, and sodium has a tendency to move down the hill across its concentration gradient. So this particular transporter or antiporter, sodium proton exchanger or NHE, 1 sodium moves inside, and in turn it just moves 1 proton outside or pumps the proton outside which is there. This is maintaining the proper cytoplasmic pH. So if there are more protons in here, the protons will be pumped out and they will be collaborating with the inward movement of the sodium. But because the direction of the movement of the 2 molecules across the transport is in the opposite direction, they are termed as the antiporters or the exchangers. Chloride and bicarbonate exchangers, or the AE, as you can see. So the chloride ion, so 1 chloride ion is moving inside and the bicarbonate ion is moving outside. This bicarbonate and chloride antiporter is very important in the systemic capillaries, and we will see that the uploading and the downloading of these carbon dioxide takes place. So what happens in systemic capillaries is there is a high carbon dioxide pressure outside. So this high carbon dioxide, it gets into the RBCs, erythrocyte plasma membrane, and inside the erythrocyte the plasma membrane, the bicarbonate is formed. So this bicarbonate then moves out and it causes the chloride ions to move inside. So excess production of bicarbonate in the cytoplasm as a result of carbon dioxide hydration reaction catalyzed by carbonic anhydrase drives the outward translocation of bicarbonate in exchange for inward translocation of chloride ions which is there. This is happening in systemic capillaries. So in systemic capillaries, the carbon dioxide is taken in and the bicarbonate is released outside. However, in pulmonary capillaries, the reversal will take place. The direction of transport is reversed such that inward translocation of bicarbonate is coupled to the outward translocation of chloride ions. So bicarbonate ions in the pulmonary capillaries, because there is a low carbon dioxide pressure outside, the bicarbonate will move towards inside and the chloride ions will come outside. The bicarbonate that enters the cell will ultimately give rise to the carbon dioxide catalyzed by this enzyme carbonic anhydrase, and this carbon dioxide which will be released outside, and we will exhale this carbon dioxide into the atmosphere. So this is the example and the functionality of a typical antiporter, which is exchanging the bicarbonate and chloride ions in the opposite direction, and this can work in both the ways, in the systemic capillaries in a different manner and in pulmonary capillaries in the opposite manner which takes place. So at the end, I would just like to point out that there are several severe inherited disorders which have been traced to mutations in genes that encode these ion channel proteins. You can see here that there are different inherited disorders. We have talked about cystic fibrosis, and this cystic fibrosis was because of the mutation in the gene CFTR gene, and which is a chloride channel, and impairment in the chloride movement was there and causing this particular clinical consequences. So any type of channels and their genes, if there are different mutations, they will alter these channels and they will cause different disorders which are there, again significantly pointing towards the function of the plasma membrane and how important the plasma membrane can be.
With this note, thank you, ma'am. Thank you so very much for giving us this session on plasma membrane. Friends, if you have any queries or if you wish to give your feedback for this lecture, then do write to us at info.cec@nic.in. We are going to meet again very soon and would be discussing on more new topics. Till then, take care. Goodbye. Thank you, ma'am. Thank you so very much.